All right, well, my name's Kara, and I'm really glad that you all came to my talk. It's a packed house. I'm going to be talking about RNA-seq data and differential expression tools in R. And where this talk came about is from my bioinformatics consulting class, and I was handed this project with sequence data, and essentially we had to do a differential expression analysis. And the first time I saw RNA-seq data, I was like, made the comical, dumb question, is it numbers? Because I realized as a statistician, I am trained a lot, and I know a lot of methods and techniques to deal with like numerical data, like with a numerical, like a matrix with numbers or an IID sample. Like, I know a lot of ways to deal with that. But in terms of the biological sciences, different data types that aren't strictly a matrix, super neat, are pretty prevalent. And sequence data is one of those things that happens all the time. And I think as statisticians, it's important to at least be familiar with this data type. And because we have a lot to offer in terms of the statistical analysis, if we can change it into numbers. So I don't claim to be a biologist, but I've been thinking about it a lot from my consulting project, and I thought I'd share with you guys what I found out. So in this talk, I'm going to talk about what RNA-seq data is and what kind of questions you can answer with it. And in addition to that, I'm going to talk about how to take the biological data and turn it into some sort of numbers that we are more familiar with to do statistical analyses. And then I'll go through a quick example of how to do an analysis of that quantitative data. And I have a short example. So we're talking about RNA. So first we're going to talk about the central dogma of biology, which is the general workflow of how genetic material, the DNA, becomes the output of what the gene codes for. So DNA is the genetic blueprint inside the nucleus of your cells, and it's the master copy that you get from your mom and your dad. And since it's like a master copy, like it wants to be protected, and we don't want to use it all the time, or at least the cell doesn't. So when it codes for things, they take a section of the DNA and copy it, and that copy is messenger RNA, or mRNA. And that mRNA can leave the nucleus and then become a protein. The process of copying the DNA is transcription. Once you have this copy, the mRNA can go into a ribosome, and then the base pairs on the RNA can be turned into peptides, and those peptides get strung into a protein. And this process is translation. And once you have the protein, it can fold the way it needs to go, hopefully, and proteins are the things in your cell that make up stuff and do stuff. So like the meat of yourself is proteins, and also functional things in your body like enzymes and the parts of the cell, organelles, etc., are all proteins. So proteins are important. So when we talk about when we're looking at RNA-seq data, like what are we trying to measure? And what we're looking at is this DNA to RNA step. We want to analyze the gene expression. And what I mean by that is how parts of the DNA, the genes, are used to make a functional product in the end. And we can answer questions such as like, is parts of the DNA, is like such and such gene being used at all? Is it being expressed? And if it is, we can try and figure out like, to what extent is this gene being used? Is it like being copied over and over and over again, which might suggest that it's important, or is it not being used very much at all? So what you would do is, let's say I'm interested in gene expression in brain tissue. So you would go into the brain tissue and get a sample of the RNA as a biologist, and you do all the careful quality control steps, and then you'd send it off and get it sequenced and figure out what the RNA is. And the output of this sequencing is RNA-seq data. So that's what I'm talking about, is like the output, the reads that come out of the machine. And what this data can tell you is the presence and quantity of RNA in a sample at a time point. So breaking that down, the presence is like whether this gene is being expressed at all, like a yes, no. You can also get a measure of how much reads, how much mRNA you found, so like a quantitative, like a quantity. And these two things are important because then you can do a differential gene expression analysis, which is fancy talk for I just want to compare the quantity or the presence between two different groups. So like I have a quantitative measure, then you can compare it and do statistical analyses on that. And another good thing about RNA is that you can look at it at a time point, which is different from DNA. So DNA sits in the nucleus of the cell, and it, for the most part, isn't changing unless like you have gamma rays or radioactive spiders or whatnot. But so like if you were to look at, say, the DNA over like puberty, it would probably stay the same the whole time. This is different from RNA, where the transcriptome is changing all the time. If you think of the kinds of proteins being produced throughout, say, like puberty, like it would be very different over time. And that can allow you to ask a lot of interesting questions, like you can look at how genes are expressed and they go on and off over time, or you could look at like maybe some event happens and you can look at before and after. So that's another interesting thing you can look at. So if you take a step back and think about it, we're really looking at the differences of a quantity between groups, which as statisticians, that's something we know how to do. We do that all the time. We can do t-tests, we can do regression models, and we can look at that. So it begs the question, like, I know how to do that with a matrix of numbers, so like, do I have numbers? Is it numbers? And if you were to look at the output of a sequencing machine like this, you'd get something that looks like this. Which, there are numbers, but it's probably not what you wanted. And it's not just the 3 entries, like you'd get like 30 to 50 million of these entries per sample. So like your data set could be very large and a lot of these things. So to understand what this is, I'm first going to talk about like what is the sequencing machine doing first. So Illumina, which is a common sequencer, goes through this process. And the key thing is that RNA isn't sequenced all in one strand. They break it up into a bunch of little pieces and then sequence each little piece. So you have all these fragments of your genetic material, and then they add adapters so that you can stick it onto like a little a flow cell inside the machine. And once it has a location, you can go through this this duplication procedure to like get a lot of them because genetic material is small. And then once you have that, you have these clusters of each fragment. And then you can start doing the sequencing procedure, which if you think about sequencing, like you have one sequence here, and like the A would have one corresponding base. So if you put the nucleotides over the mixture, like the T would match with the A and such and such. So as the T binds to, say, like one of these, it lets out a little fluorescent signal, like a little piece of light. And this machine can look at all of the light being emitted from not just one cluster, but all the clusters at the same time, and keep track of, well, this blue light is coming from here and that's the first one I saw, so therefore the first one is an A. So when you're looking at the output, you're going to have a sequence not of the whole thing, but of a bunch of fragments. And there is this uncertainty of like, I'm pretty sure I saw blue light, but it's not 100% sure that it's blue light. And so there's a degree to like what base pair it thinks is in that sequence. So if you look at this again, first you have this, you have the GCs, A's, and T's, and this is the sequence of the base pairs of the fragment, of one of them. It's called a read, just one piece. And then above it, you have all these letters and such. And these tell you exactly where that cluster of fragments was on the tile. So it's like the machine number, and then the flow cell, and then in the flow cell the tile, and in the tile the coordinates. So if you think something went wrong on some location, like maybe that could be useful. And then there's also this gibberish, which is letters, but not base pair letters, and also symbols. And these are the quality values of the sequence. So there's one symbol for each of the base pair letters above it. And these indicate like how confident it is that, say like, if that one base is a G. And the clever way that they did it is each letter corresponds to an ASCII number, and there's a quality score between 1 and 60, and then there are 60 ASCII numbers. So in general, like the letters are better quality than the numbers, and I think there's symbols on both ends. So you'd have to look it up, but like if you thought something went wrong, it would be like, you could look and see how confident it is.
In this field, did they ever do like a zero rate of negative binomial and then don't filter on the database? It's not done for genetics?
That's a good question, and I don't really know what the like norm in the field is. From what I gather, that they do take out the zeros. I'd have to look around to see what the consensus was, but where I learned to do that is from like an article in Nature, so I assumed that was something that was done a lot, but I'm not sure if other people do different things.
And one other question that I think somewhere in the code, you did the first model matrix. I think it had a no intercept. Is that?
Oh, it was like 0 plus group.
So that's common to put a model out of it. So that's what that function.
For that one, like I put the 0 in there because later I wanted to do a contrast with like the 3. Okay. So like you could do it the same like without, just like your contrast would be like different numbers. I just thought it would be more clear because I said like 3 groups and then like there were 3 numbers there. But definitely like you could do it without it. You just have to change what like your syntax later on was. Mm-hmm.
A couple slides ago, you reported a likelihood ratio test and the p-value associated with it. What's
Yeah.
the null of that test? Null hypothesis? Does that have something to do with whether the gene is expressed or to what degree it is expressed?
So the specific null for that would be like in the likelihood ratio test, whatever you said to do. So like if you were working with the regression model and you were saying like coefficient equals 2, it would be like for that parameter equals 0 in the negative binomial regression model.
Okay.
So are the genes like multiple response variables, or do you have a coefficient in the model for each gene, or
So you can actually do a test for like each gene. So like there is a sequence of counts, and let me go back to the data. Yeah. So like there's one gene and then a sequence of responses based on however many samples you have. And like I imagine like you could have this go farther and you'd have more than like 6 or 7 that I put on the slide. So the test is for just one gene.
whether it's different between groups?
Mm-hmm. Yeah.
You talked a little bit about how certain packages are faster than other ones. How much is that a concern? How much basic does it take to
So, like, how big are the data sets, and like, is it important for it to be fast?
Like, did you run into speed issues?
We did run into size issues. Like, the speed issue is really how fast you need your results. So I think we estimated, because we had, I think, 40 million reads per sample, and then we had 64 samples. So like, we estimated we could get it done in like half a day or something by using the cluster. But like, if you didn't have a cluster, or if like you're already like kind of straining on the data set, or maybe if you had like a lot more data than we did, then maybe that could be an issue, because these data sets can be big.
I guess my follow-up to this question. So this is sort of like the data set that goes into the model, so our observations are genes, or those our observations are genes. Is that the right way to think about it?
So like, you're doing a test for the gene, so like each one. So like your like samples would be like your observation. Like, these are like all the Y's in like one row.
In the multivariate data, the sample being in a unit. One sample is the same data set, and there's data for each gene in for each unit.
So you're saying, can you phrase that again?
Which one's the sample unit? Is it the gene, or is it the sample that's mashed together brain cells?
So you're looking at each gene separately, yeah. So like, you're doing one test for each gene, which is why I was talking about the multiple comparison thing.
So when you fit the model, it's actually fitting a model for each separate gene, and then you're just putting all those P values and then you're ordering them as they fall.
Yeah.
In the columns here, different fish types or different parts.
Yeah, so like, I took this from a paper, so I'd have to look up what the CT and PA mean, but it would be like, maybe one would be treatment C, and then it was on line PA or something. Like,
But I can think of the columns here, like different people.
Yeah.
Okay.
mm-hmm.